Okay, so we are going to pick up where the last video left off, just talking about the parts of the nerve cell. And so looking at this particular slide that you guys have, you get a very good visual of the difference in what the nerve cell looks like compared to what your typical body cell looks like. So I'm going to talk you through these parts, and we're going to basically start at the top and just work our way down. So the first one that you have on there are the dendrites. And the dendrites are these extensions that you have off of the nerve cell. And this is actually going to be where information comes into the nerve cell. So dendrites are going to bring an impulse in. That's their job. And the reason that you have a variety of dendrites all around that cell structure is because you never know exactly where an impulse is going to come from. And so by having those extensions coming off of multiple places, it just allows that cell to be able to pick up that information no matter what direction or where it's coming in from, so that the nerve can start processing that information. So dendrites bring the information in. That's their job. Just below that, you have the nucleus and the nucleolus, which should not be new terms. We talked about these in our cell chapter. So the nucleus is the brain of the cell. It is going to monitor what's going on in the cell and make changes as needed. The nucleolus is a small dense region that you have inside the nucleus. And when we initially talked about it, I told you guys kind of think of it as an egg. Nucleus is the white part of the egg. The nucleolus is going to be the yolk of the egg. Now you'll notice the speckling appearance around the nucleus, and it's referred to as the chromatophilic substance or Nissl bodies. Now what these actually are is these are structures that you guys have within the nerve cell that are going to help the nerve cell produce protein. Protein is something that we use all over the body in pretty much everything. It's in our DNA, it's in our muscle cells, our skin, our cells, membranes. Um, you see it all over the place. And so because the number of places that we need to have protein, you have these special structures that are going to help you produce protein all around the nerve nucleus. Now you'll notice the bracket there as well, which shows that core structure of this particular nerve. That is the cell body. And the cell body, if you were to put a mark around the cell body and not include the dendrites on this, you could very easily see that basic circular structure that we kind of think of ourselves having. So it is there at the very core, but you have all of these added features so that the nerve cell can work the way that it's supposed to. So the cell body is the main part of the nerve. Now, if we move down the cell body, you'll notice that it starts to taper. And so we get this narrow section at the bottom of the cell body. And we have two structures there. One is called the axonal hillock. The other one is a neurofibril. Now, the axonal hillock is simply the transition point between the cell body and the axon. So that's all the axonal hillock is. It's where it tapers, starts to narrow, and it turns from the cell body to the axon. In that same area, you have structures that are known as neurofibrils. And you can't really see them here, but they are rod-like structures that run through that narrow section. And the best explanation I can give you for the neurofibrils is they are essentially lightning rods. So if you look back at those dendrites, you have these impulses that could be coming in from so many different directions. And when those impulses come in to the cell body, they have to be collected, they have to be kind of ordered so that the nerve can actually do something with them. And so in order to get that done, these neurofibrils are essentially lightning rods. And if you think about what a lightning rod does, if you put a lightning rod on top of a building, you're trying to pinpoint and get all of that energy from a lightning strike collected into one central location so that it can be dealt with in an orderly fashion. And that's what the neurofibril is trying to do. It's trying to take all of those potential impulses that are coming into the nerve cell and get them ordered and collected so that the nerve can actually do something with them. So once those impulses are all collected, now it's going to travel down this long extension of our nerve cell, which is known as the axon. So the axon is very narrow, typically the longest part of our nerve cell, and it's going to help direct the impulse down the nerve as a whole. Now on the axon, you have the potential to have a special structure. It's not on every nerve cell. They show it here, but please understand that it's not on every nerve cell. You have a special feature which is known as myelin, or the myelin sheath. And the myelin is a protective covering that you have over the axon, and it's a protective covering that's made of fat. And if you look at your next slide after this, it talks about myelin as well. So you can kind of be looking at both of those slides at the same time. But myelin is made of fat, and it is going to offer protection to the axon, and it also helps speed up a nerve impulse. So if you really look at the picture, you'll notice that the myelin sheath is not continuous, that there are breaks or gaps in it. And if you look at the labeling, you'll notice the nodes of Ranvier. The nodes of Ranvier actually are talking about those spaces or those gaps that you have between the myelin. So wherever there's a break in the myelin sheath, that area is known as a node of Ranvier. Ranvier is actually the last name of the scientist that discovered these, and they are exposed sections of the axon. Now, if you think about this, when we talk about the nerve impulse, and I've mentioned it several times already, it's basically an electrical impulse that is being sent through your nerve cells. If you think about what are great conductors of electricity, a couple of them probably come to mind right off the bat. And we know that metal is a great conductor of electricity. But on the flip side of that, if you think about what are very poor conductors of electricity, rubber would be one of those. Now, we don't have rubber in our bodies, but we do have the equivalent of that, which is fat. So if you think about that, initially it sounds like a contradiction. I've mentioned several times that the nerve cell is designed for speed, and now all of a sudden we wrap it in something that is designed not to conduct an impulse very well. But here's what actually happens. So as that impulse starts to move down the nerve cell and it runs into those bundles of myelin sheath that are made of fat, it doesn't continue to move through the axon. It will actually jump over that section. And those exposed areas, the nodes of Ranvier, that's where it will jump to. So it jumps over the fat to those exposed areas and basically hops its way down the length of the nerve until it gets to the end. Now, think about this. If the next time we were in class, and I'm not going to make you do this, but if the next time we were in class, I had you guys all get up and stand shoulder to shoulder. So you're standing next to each other, shoulder to shoulder, and I gave a ball to the first person in the line. And I said, okay, you have to pass this to the next person. They'll pass it to the person next to them. And everybody has to touch the ball, and we'll see how long it takes us to get to the end of the line. Okay, we're roughly 28 people in class. So all 28 people have to touch the ball, pass it on to the next person. The next time we do it, I hand it to the first person and I say, okay, now when you throw the ball, you're going to throw it to number 14 in the line. Number 14, you're going to throw it to number 28 in line. And we get to skip all the people in between. Well, which one's going to go faster? Obviously, the one where they get to throw the ball and skip a bunch of people in between. You're going to get that ball to the end of the line much quicker. And so that's essentially what your nerve cell is doing. Those nerve cells that have myelin are able to jump certain parts of the nerve cell. And so they can get the impulse from the beginning to the end of the nerve cell much faster. Now, the presence of myelin, fat has a light color to it. We typically think of it as being kind of a cream color or a yellowish color, but any of your nerve cells that are wrapped in myelin are actually known as your white matter. Those nerve cells that do not have any myelin and they have the exposed axon, your nerve cells are actually gray in color. And so that's gray matter. So when you hear those terms being used, white matter and gray matter, white matter indicates that those nerve cells are bundled with myelin. Gray matter indicates that they lack myelin. Now, your myelinated cells, because they get to jump to those nodes of Ranvier to make the impulse or get the impulse down the length of the cell, they work a little bit faster than your unmyelinated cells. Not that your gray matter is slow by any means. We're still talking about split seconds to be able to send information, but the myelinated cells are going to be able to go just a touch faster than what your gray matter will. So the myelin sheath, like I said, we were kind of talking about those two slides at the same time. The last couple of slides talk about the neuroglial cells. And you have some neuroglial cells that are specific to your peripheral nervous system, and you have some of them that are specific to your central nervous system. So when you look at the neuroglials of the peripheral nervous system, you have the Schwann cells and you have the satellite cells. Now, Schwann cells, again, this is named for the scientist that discovered it, because the cool thing about making a science discovery is that you get to pick the name. So if you find something or discover something in the world of science and you want to name it after yourself, you are completely able to do that. And so that is what these scientists decided to do. Nodes of Ranvier. Ranvier named him after himself, and same thing with the Schwann cells. They were named for the gentleman that actually discovered them. These are the cells that are in the peripheral nervous system and produce that myelin covering. So if you are talking about Schwann cells, you know that you're in the PNS, you know that they are going to develop that fatty covering for your nerve cells. Satellite cells are a particular type of cell that are going to help what are called ganglia. Now with the nerves, most of your nerve cells are going to have the dendrites, the cell body, the long axon, but not all of them are constructed that way. Some of them have just the dendrites and the cell body. They don't have that long axon extending off of them. So if you have just dendrites and cell bodies, they tend to get clustered together, and those are called ganglia. The neuroglial cells that you have in the CNS, we have a couple more here, and that's in part because of the job that your brain and your spinal cord have to do. First one is the microglia, and they get that name micro. They're the smallest ones, and that prefix is just indicating their size. These are cells that are actually going to eat anything that is building up around the cell that is debris, could be potential invaders, bacteria, things like that. So they are just maintaining the conditions around the nerve cells to make sure that things are not building up, it's not becoming cluttered around the nerve cell. Then you have the astrocytes. Astro indicates star, and so these actually have a star-like appearance, which is where their name comes from. And these are the cells that are going to help produce scar tissue. So if you do have damage to a particular nerve cell, it's going to be the astrocytes that are going to help and try to rebuild and repair that so that the cell doesn't die. They also are going to help form what is called the blood-brain barrier, and a lot of times you'll see this abbreviated as just BBB. The blood-brain barrier is something that is going to help the body regulate what is able to move into your nervous system. Because of how important the nervous system is, you don't want the wrong things to be able to move in and out or across those cell membranes. And so the blood-brain barrier is just kind of an added level of security to make sure that the wrong things are not able to make their way into the nervous system. Still can happen, but it just helps decrease the chance of that. Number 3 on the list, the oligodendrocytes. These are going to be the cells in the central nervous system that are going to produce myelin. So you have a different class in the PNS than what you do in the CNS. They're going to produce that fatty covering for your brain and for your spinal cord. And then the last one, the epidermal cells. These are going to be the cells that are going to help with your CSF, which is the cerebral spinal fluid. And this will be coming up in our future talks as we move into the brain and the spinal cord. But cerebral spinal fluid is essentially a fluid that you have in your nervous system, constantly bathes over your brain and your spinal cord to help make sure that it's getting all of the ions that it needs, that it's getting all of the nutrients that it needs, and just to make sure that the system is staying as healthy as it possibly can so that it can function at the highest level. So the cerebral spinal fluid, it's constantly getting flushed over the brain and the spinal cord, and it is being drained off, and new cerebral spinal fluid is being put in so that you are constantly getting the cleanest and best fluid that you can through those two structures. The last slide that you guys have talks about the ions of the nervous system. And there are two major ions that we have when we talk about our nervous system. And these are really going to be the ions that are going to be responsible for that nerve impulse being able to fire. So the two that we have are potassium and sodium. And what happens is you have potassium and sodium, they're going to be on opposite sides of your cell membrane. So you have that tube-like structure through the middle. That's essentially trying to represent your cell. And the outside of that tube would be the cell membrane. You'll notice you have a row of circles on the outside of that cell membrane, and then you have a row of circles on the inside. And that's essentially how your nerve cell is set up. You have these ions that are on opposing sides of the cell membrane. And when an impulse comes in, these gates on the cell membrane open up and they allow for those two ions to be able to interact with each other. Now, as those ions interact and start changing places with each other, that is going to be what starts the current for that electrical impulse that we were talking about earlier. So potassium and sodium, two very, very important ions in your nerve impulse. They are always kept on opposite sides of the cell membrane until you actually need the nerve to fire. Once you need that nerve to fire, the cell membrane is going to open up, have these special channels that will open up, allowing for those ions to switch places with each other. And just that switching, that movement of those two ions, because remember, ions carry a charge with them. You get those in motion. Now you create that electrical current that is actually going to fire that nerve impulse. So that is our introduction into our nervous chapter. And we will pick up and talk about the brain and spinal cord as we move forward with our next discussions. Have a good week, and I will see you later.